All right, well guys, welcome to the cropping matrix once again. I'm here filling in for Dr. Randy Kuchar from the University of Saskatchewan, who couldn't be here for particular reasons. And I'm going to show you this giant trial once again, except the exciting part is we're on year 3 now. So this has had 2 previous crops already grown on it, and year 3 is where all the magic is going to happen. So you can see George and I were kind of playing around scouting some of the disease there before we got rolling. And I'm just going to back things up and start off on Fusarium because I like to frame the talk on Fusarium and just kind of review all the basics because I think that's the framework for where all the other pieces that we're going to see fall in. So Fusarium Head Blight, FHB, that's become a problem in Western Canada for a while, but its onset has been a little more recent. And this affects a lot of our major crops and important things. So we've got our bread wheat, we've got our durum for pasta, and even the beer. The beer guys are really picky about quality for a product that's been grown out of river water and stuff on the ground for 2,000 years. They're really particular about quality specifications. So we've got a major problem that can happen when we have Fusarium in our crops. So we've got a number of different treatment options. What are the main ways that we prevent FHB? I got some experts. I'll try and ask some non-experts here, like our summer students if they're around. What are the main ways? We've got fungicides are an obvious one. Watering schedule, that's been one that we've looked at in some previous projects, and we'll talk about that. We've got resistant cultivars, I don't know if I mentioned that. And then we've got our rotation, and that's where we're going to get to. So this study specifically, this is, I'm going to skip ahead I think here. I'm skipping way ahead. All right. Maybe I'll back up then. So when we're talking about Fusarium Head Blight, what happens is it produces Fusarium damaged kernels, and we have a mycotoxin that's produced within the kernels. And so this is spread in a number of different ways. So one way is by bedding. You can spread the spores. So the thing about bedding though is it's not a major problem, and that's because if producers are careful about how they're using their bedding and disposing of that sort of thing, it's kind of killed off by compost. Another major way that you could spread Fusarium is by the straw, so if you're baling and feeding to animals. But again, that's going to be killed in the gut of the cows that it might be fed to, so that's pretty minimal. We've also got airborne, so spores are airborne. We're going to talk a little bit later about some spore trapping projects that we're doing. But how far can the Fusarium spores fly, Mike? Like a few kilometers at most?
Most of them would be meters.
Okay, meters, so even less.
A few of them might make it to the next field.
So I've heard that maybe a kilometer or 2 is most, but Mike's saying like even just the next field. But that's really low percentage of those spores. What's important for Fusarium is that it's a residue crop? A residue-borne pathogen. And so what that means is that the pathogen is born on the residue from the previous crops. So it's not like a leaf rust or something where things blow in from North Dakota and Minnesota and they come up through Manitoba and the major air currents move them here and cause a problem. I mean, this is really a local problem that we have if we have it. And so why do we have it? Well, probably one of the biggest reasons was Fusarium infected seed or using damaged seed and bin run seeds through the 90s and 2000s, I suppose. And so that's where it comes from, and it's in our area now, and so we have to deal with it. Obviously, some of the other important things to think about is just if it does become a problem, you're going to have lowering grade at the elevator. You're going to see feed decline, possibly refusal from animals it's being fed to. And the tolerance levels at the elevator, there's an entire, obviously, CGC guide on that, but we're talking like 1 to 2 parts per million. And I'll share some results from what we got in the fields here later on. So genetics, if I kind of talked about those 4 things, the rotation, the genetics, fungicides, and other cultural practices, where would you guys rate genetics? Any breeders here? George says at the top. All right. Is that in general, or is that specific to Fusarium? Okay, in general, you're saying genetics way at the top. I don't disagree with that, but I think when it comes to Fusarium, that might not be the case. And that's because, like we said, it's born to the residue. And so genetics are only going to help so far. Now when I was looking into this, there's some genes that have been going strong against fungal resistance for like 50 years now, which is pretty incredible. And you can compare that to other things where, I forget his name, some genes out of the University of Alberta in different crops, and they've lasted a couple of years, or maybe 5 or 10 years before that resistance was overcome by different pathogens. So it's not the thing I'd want to hedge my bet on, I suppose. So then we've got fungicides. Where'd you rank those? For Fusarium? Yeah, it's probably up at the top, but the challenge with fungicides is a lot of producers don't necessarily want to use them. They're an insurance policy, but talking to some of the guys from our plot hop last week, economics aren't always there for them, even if they are paid for. It's a tough sell depending on the logistics. So, and then we've got the cultural practices. So George mentioned that when I first started, probably 10 years ago, we had a Fusarium project, and we went around all the different fields and we scouted at different timings. And one of the things we looked at was that irrigation timing and stopping irrigation during flowering. And the thought being that if you can lower the relative humidity and the leaf wetness, you're going to have less chance of those spores kind of floating around and infecting the flowering wheat head. So the challenge is we're in southern Alberta, obviously, and you really are going to have to make sure that you top up your water levels as much as you can before you get to that flowering timing. Because if you don't, you really don't want to stop watering by an inch or two over a pretty critical week window or so. So that is a challenging practice. I'm not sure, has it been adopted by many folks around here at this point? A few? Yeah. And are they finding it's pretty effective? Yeah, it's hard to say without a research perspective. It helps them what? It does. I guess it's just like the fungicides. It's a pretty good insurance policy. If you know you're following a practice like that, it's going to be really good. So then our fourth thing, rotation, rotation, rotation. And I think that's where the magic of this study behind me is going to come in. And it's kind of being argued within the study that that's going to be the most effective means of controlling Fusarium. So obviously, if we're talking rotation and you talk to a researcher, they're going to tell you, like, you can completely eliminate Fusarium if you just don't grow any host crops for 10 years or 20 years or whatever it is. But the reality is that's not the economics of farming. And that's a challenge that we're up against, is how do we balance these best practices versus the economics that we know are going to be in play. So, well, interestingly enough, when I was looking into this, I had an old AgriFax with me, and back in the 2000s, I'm not sure they had much for gene resistance for FHB yet, but now they're looking at genetic solutions for FHB right out of North Dakota, FHB 7 gene, and they're hoping this is available within the next couple of years. So maybe they will pop up that the gene side a bit. So timing 2 to 3 weeks after flowering, that's when we're going to see the symptoms start to develop on the head. And so there's a couple forecasting tools we can also use to look at this. So the Alberta Wheat Commission, in part by Ralph Wright and Pavel with ACES, that's the Alberta Weather System, and Michael Harding and Neil Watley from ARD, and Kelly Turkington from Agriculture, Agri-Food Canada. So there's this Fusarium risk tool, and I actually played around with it a little bit this morning, and it said, right now our risk is low. And that's because we've passed this flowering phase already. Further to the north of us, near Wetaskiwin and a few places, there was some bright red kind of lines there showing they're really high risk right now for Fusarium head blight. If you were to go back a couple of weeks, we were intentionally irrigating these plots, hoping to kind of produce a bit of infection for research here. So we'll see in a week or so how effective that was. And also we're working with Dr. Manasseh McBalla out of the University of Manitoba. And he's doing a similar study, not to this, but a smaller plot study. And he's integrating the spore traps and weather stations. And so Lewis might have a bit more to comment on that, the spore trapping side of that later on. And that's based off a USA approach. And they figure it's a pretty good approach of assessing the forecast of the Fusarium population. So to get to this study, it's going to be a challenge to walk you through, especially with the online folks. So I'll probably point you in a few different directions, and then if we have time, I'll take the crew through after and we can look at some more specifics. So this study is actually, it's in Lethbridge, but it was up in the Peace, in Falher, in Melfort, Saskatoon, Indian Head, Brandon, and Morden, Manitoba. So all across Western Canada, and targeting different growing conditions, different crops, different host species. So it's going to be a pretty complex and robust kind of data set in the end. In Lethbridge here, we're looking at wheat, durum, and barley as our main cereals, corn as well. And then we've got canola, dry beans, pea for peas. And we threw hemp and quinoa in as part of another study. So some results that we saw over 2018 and 2019. So we've looked at a few different things. We've looked at the leaf rating. So we've got a rating scale for our flag leaf and penultimate leaf. And we're looking at the amount of leaf diseases on those. And then we're looking at an overall disease score as well as the Fusarium damage, and taking samples in and looking for Fusarium damaged kernels as well. So when we did the Fusarium assessment, I got something down there. I think I got a bug down my shirt. When we're looking at the Fusarium assessment, there's an incidence and severity score. I'm kind of far away here. I'll find it. It's a Stack and McMullen. And all we're doing is we're looking at a head and seeing if we can find any whitening of the kernels. Usually if there's kind of a fluorescent pink or orange in there, it makes it really easy to know and confirm that you've got Fusarium damage. And so we'd go through each one of these little plots here, assess 100 plants, and see whether or not we've got Fusarium. And then based on the severity of that, you come up with an index for FHB. Now in 2018, that was the first year we did the study, so we would have just had big long blocks of wheat and barley and peas, and some sites had oats and other things. Our incidence and severity was pretty low. 2018 wasn't a huge year for Fusarium, I don't think, across the prairies here. Melfort had like 1.2% index, and most of the rest of the other sites had less. Although we still detected species within the samples when they were taken to the elevator. I think some of ours specifically only 15 parts per billion of Dawn, so well below the threshold in that first year. But really the magic is coming from stacking the crops on top of one another, and then this third final year, hopefully growing durum, seeing a little higher, unfortunately higher Fusarium. So the other thing we're looking at is the leaf diseases. So we're taking a flag leaf and a penultimate leaf down below and assessing whether or not the crop rotations have an impact on these leaf diseases. So when it comes to the flag leaf, that's where you're going to have the biggest yield impact. And the theory is that if you've got a really high infection on your flag leaf, you're going to reduce your yield. So we wanted to see, based on the rotations, whether or not that could be affected. Any thoughts on if that actually showed anything? You've probably done a lot of this work, or maybe seen no guess for me even? It's hard to say.
It's sometimes tough to tease out the differences between climate or weather
Yeah.
and the rotation. So it's hard to guess.
It's hard to guess, yeah.
In years that it's dry, it may not have a big effect, but in years where the disease potential is high, I bet you'll see the difference.
Okay, so Mike said he thought we'd see the difference when you have big disease potential, maybe higher yield potential. And I think I agree. We've only got, obviously, the first 2 years, not the third. We did see barley on barley, these penultimate leaves, the second from the top. They had an increased leaf disease from barley, but only a few percent higher. There's a pretty robust data set, so we're quite confident that there is a thing there. But I think maybe it comes down to those disease cycles in this case. If they are blowing in from other places and infecting the plants, those other factors, I guess the genetics of the variety that we chose and other environmental conditions probably have a bigger effect in that case than the residue. And that's in pretty big contrast to the Fusarium. So I guess one of the other things to mention is we planted corn in these trials. That was pretty intentional because Fusariums, or corn is a host for Fusarium. So we've collected cobs and we're going to send those in and see whether or not we get a really good infection that way. Is there anything else I should touch on in regards to Fusarium, or are there any questions? Yeah.
I was wondering if you're doing a lot of irrigating leading up to flowering, would that help to release more spores?
The question is, if we irrigate a lot leading up to flowering, or even as like a technique for being able to shut off maybe at flowering, are we going to increase the release of spores? I don't know. I'm going to defer to the expert Michael, and he says maybe.
Definitely
Maybe. Yeah, definitely might could. That's a good question, actually. Like maybe throw that back at Randy and see.
I think irrigate to the needs of the crop first. If you can shut off during anthesis, do that.
Yeah, so what Mike's saying is irrigate to the needs of the crop. And if you can afford to shut off, then awesome, you can. Yeah.
Have you put any monitors out there like last year when it was dry? How wet it is in there on a morning like this morning? It was wet this morning, didn't it?
That's a good question. If we put monitors out there, no, we didn't, but maybe we just have to do that boot test. Like you said, it didn't rain. My toes are soaking wet through my boots right now, just walking through. There's a lot of dew, and I'm not sure it feels as humid as I'd expect, but it's pretty, even though it's pretty hot today, I think it's not like a Ontario muggy right now, but it's wet. There's dew all over the ground all the last couple of weeks, so hopefully that'll help. How am I doing for time?
The other question,
Yep, okay.
this goes back to chickpeas. Now you carry this crop, take it off, and you see chickpeas in there. How much is that going to affect the chickpea crop?
Okay, so the question is, say we're done this study and we fill it all to chickpeas, how much is the Fusarium and other hosts going to affect the chickpea crop? Good question. I'm going to defer.
Yeah,
What do you think?
Fusarium is a tough nut to crack because some of those species that'll be on the durum are going to be able to infect the chickpea. And so probably not Graminearum is going to be your biggest problem in chickpeas, but other species like Redolins and Avianaceum, so that whole species complex chickpeas. It'll cause root rot and crown rot in your peas. It's a tough one to get on top of. And if the previous crop leaves in the residue a lot of Fusarium potential, then yes, it certainly could. If the crop you're going to put in is susceptible to Fusarium,
Yes.
it could be a challenge.
So the question was, again, if we seeded chickpeas over a Fusarium area, what are we expected to see? And yeah, not specifically Graminearium, but some of the other species may infect those crops.
Probably redolent.
Redolent on chickpeas. And it's going to be hard to tell the difference. So that's one of those complicating factors. Let's see what I have left there.
Then do all the
Yeah.
fungicides get every one of them?
Do the fungicides take care of that? Each species? Well,
Resistant to it, or they won't get it, or not?
There might be some minor differences, but the label should tell you which species specifically have been tested and demonstrated to be
yeah,
efficacious against. But yeah, you can't necessarily count on every species of Fusarium responding the same way to a fungicide application.
yeah. So to go.
It's not on the label, don't assume.
Yeah, so to zoom back out, we're talking about Fusarium in general often, but like Michael said, it really depends on which species it'll control. And in this case, Fusarium graminearum is the key one. There's also Culmorum that we've seen at higher densities within our plots. And they measured the different species from all the samples, and they were quite variable between all the different sites that we had. So it definitely varies from location to location. So yeah, that's maybe another reason why fungicide might be effective here and not somewhere else, because it's, even though you think it's just Fusarium, it's an entirely different species or that sort of thing. And the other thing is, from what I understand, not to rely too heavily on fungicides like what am I thinking? Try to be effective, but some of the strobulins and others aren't as effective, and we've seen some crops or some diseases and pathogens kind of overcome those fungicides. And I'm not sure, chickpeas, the pathogens in chickpeas like Ascochyta kind of overtaking some of those fungal products. At this point, I think it's kind of safe to say that, that's why it's important to have all those different strategies.
Just to be clear, the triazoles for Fusarium Head Blight are registered for suppression only, not for
Okay,
control.
yeah,
So
and
you can't expect to control Fusarium Head Blight with triazole fungicides.
And-
because they can suppress, but if you're growing wheat on corn stubble, you might not be able to suppress it enough to avoid yield
okay. So
downgrade.
the comment was that if you're growing, say, wheat on corn, you're not going to be able to suppress the Fusarium enough just from the fungicide alone. You may still see yield damage, just not necessarily as much of a decline. How am I doing for time? I'm just thinking about change in pace here. So if I got another 5 or 10 minutes, we'll get into the rotational side of things. That's where we can get moving. So let me find my map. I'm in. Well, actually, one other fun thing I wanted to do while I had George snooping around the plots was kind of pull out some of these durums and show you some of the leaf diseases that we have right now. So if I can get a couple volunteers, and actually I wouldn't mind, George, if you took that plot. Yeah, Michael, if you want to do a one beside him. Does anybody else want to come do one beside me here? Maybe we'll see if that theory holds true. Because we've got a different year one plot down each of our strips here. And then, so we've got quinoa's growing there in year one, and dry beans were there in year one. And then all down this first strip, we had peas grown last year. So peas isn't a host crop. We'll expect the disease maybe to be lighter, but we'll see. Scott's peeking around there. What are you guys finding for disease? And we can figure out what we want Mike to talk about after lunch here. I'm going to bring up some of my diseases for you. So this is all Spitfire durum. So it's got a relatively good disease package. And one of the problems with doing this study is we almost want crappy seed, but it's hard to find because we've got so many seed growers and certified seed grown in the area that it's hard to find crappy seed to do trials with. And most farmers aren't using crappy seed either. So here's kind of a look at what we've got. These are our flag leaves here. There's a lot of powdery mildew on those penultimate leaves. Not so much on the flag, but. Were you seeing stripe rust on some as well? Mine don't really have stripe rust. You bring some more over. So every second plant with stripe rust in it? Oh, you can show the camera.
Look at that, even some
Go show the
grasshopper damage.
camera. Grasshopper damage. So that complicating factor is we've definitely seen some cereal leaf beetle maybe, some big grasshopper damage, and some other things. So training the crew up on doing these ratings is pretty fun. Yeah, up to the penultimate with powdery mildew. Same with mine. Yeah, they're quite infected.
One reason I don't like working with durum, it's such a fussy crop.
So we've got quite a bit of disease going on here. I was kind of hoping we'd have a few more people and just get you guys to help me do the ratings completely, but that's okay. And Septoria, yeah. Hey, keep them spread out. You got to be the COVID police here.
Same thing. The one interesting thing is that because stripe rust is an airborne disease, it comes and settles in the upper canopy, and that's often where you see it first.
Yep.
Whereas these other residue-borne diseases, they start at the bottom and work their way up.
So like powdery mildew?
So the powdery mildew and the tan spot and the septoria are coming from the bottom up, and the stripe rust is kind of starting from the top.
Yeah, so where we see stripe rust is just starting on those flag leaves, and as you get further and further down the canopy, we are almost completely taken over on some like the lowest leaves with powdery mildew and tan spot and septoria, things like that. So we're mainly focused on the disease, but you can see some really other interesting things going on here, and that's going to probably play into our end results. So I'm just going to show you a couple more things quick that haven't shown up yet in years past. Have any of you guys been down to this far plot, the very corner? Well, everybody's in the disease world now. Hey. Gotta be the police. So I get really excited when I go past that very corner plot, and it's probably going to be a nightmare, but it's lodged, which doesn't happen usually in our small plots. And when I dug open my map here, we were doing some ratings and scouting, and that's where we've had peas on peas for 2 years and then planted it to durum. So we've probably got a little bit higher nitrogen there, potentially higher yield to the point where we've actually seen lodging. And then there's a couple other plots here that started to lodge as well. Those were on dry beans before and then peas after, and followed by durum. So some interesting things like that are starting to show up in these plots, and you really don't see it until you get to this here. So it's pretty exciting. The other couple of things you might see is we've got some wild oat problems. And if I were to take a map and give you an aerial view, most of our wild oat problems are coming in a couple particular crops. Not surprisingly, I should say, are anywhere we've had hemp and quinoa the last couple years, we've got some weed issues. That's because the in-crop options are pretty limited. You can put edge down ahead of time for hemp, and you can spray a Sure or something like that in crop, but for grassy weeds, so we should maybe do a better job, but still quite weedy in those plots. And the other places are cereals. So wherever we've had Durham on Durham, or Durham, so coming, if I start coming down this way, you'll notice this had Durham in year 1, and so there's some clean plots, and then there's some pretty weedy plots with wild oats. And where there's weedy plots with wild oats, that's where we've had cereals, cereals, cereals. And where there's plots, we had peas here, so they would have got a Sure post, and then we had quinoa beside that, so it would have got a Sure post. And then down where I've got the flag there, it would have been Durham on Durham. So that's the group 1 and 2 resistance challenge. So we're here trying to study Fusarium, but all these other things just always pop up, and they're pretty interesting. Our wild oat plot there is kind of the same thing, Durham on barley. So how am I doing? I think I'm pretty much wrapped up, unless there's any more online questions, or if you want to try and hopscotch through the plots there. I've got a lot of good slides and visuals and rating scales, so if you want to re-watch any of this, I think Morton's a whiz at popping some of that stuff, and maybe even get an aerial photograph of this stuff put in on the slide so you can go back to it later. And you guys are more than free to wander through if you have any questions, or you want to scout for some diseases later on and stuff. That'd be lots of fun. Can Fusarium cause leaf lesions? Is that the question? Not that I know of. Fusarium mainly is going to affect the kernel. That's where it comes in at flowering. It affects the kernel. You get those really dented kind of kernels. And as Mike says, root rot, crown rot, head blight. For what? That's Fusarium. Okay, Fusarium will cause root rot, head rot, and crown. And actually, that's a good point. I forgot to mention that. That's another assessment that we're doing, is coming in and digging up plants and then looking at root rot. And root rot's got a really great scale from letting them to assess whether or not it's damaged. It's basically clean or slight, moderate, or heavy. Really, really simple to do. So, any more? All right. Thanks a lot.
Hey Mike,
Yeah.
what about seeding rate?
Oh, seeding rate. We're not looking at seeding rate in this study. I think we seeded to 250 seeds per meter squared, like pretty typical. So I should mention that, yeah, for the most part, our cultural practices are pretty standard within this trial. I think it was 250 or 275. Can't remember offhand. You know, I think it was 10-inch spacing. Yeah, the biggest challenge was kind of the sourcing the seed and trying to figure out, you know, we really want to use something that growers are using. You don't want to use a seed from the 1980s and prove a point that doesn't prove a point. That's no longer relevant, right? But at the same time, it makes a difference when your seed is naturally, or not naturally, I suppose, but it's resistant to maybe some of the rusts and things like that. Yeah.
One thing you didn't talk about when you talked about different ways of managing disease was tillage.
Well, tillage. Well, I forgot. I said cultural, but I forgot about tillage. And tillage is actually a really good point, because Fusarium is residue bound, and tillage is one of those potentially good ways to get rid of it. I mean, I suppose what tillage, the theory behind tillage is what that's going to do is it's going to cycle that straw and break it down quicker. Does that actually work?
So would it delay it, or does it actually reduce the amount of spores in total that are produced?
Would it delay or reduce the number of spores? Well, I guess what the tillage is doing is it's mixing up the soil and the straw and hopefully breaking it down faster so it doesn't just sit there. I'm kind of I'm from after zero tillage, so I don't remember the days of
Yeah,
wheat
with
fallow.
residue-borne diseases, you want the breakdown of the residue to occur as quick as possible.
Do you or Mike have a recommendation for rotation to prevent aphanomyces?
So the point was the tillage is going to help break it down a little bit faster. Is there a recommendation for Aphanomyces? Yeah, Sham has been working on this problem, Shama Chatterton from Ag Canada for quite a while. Is there a rotational aspect that's working? 8-year break? Is that realistic?
Yeah,
What do you think?
unfortunately, we don't have any good solutions other than if there's bad aphanomyces, don't grow peas or lentils there.
Yeah,
So field selection and crop rotation. How long is long enough? Depends on the field.
so yeah, the best answer I have is probably the best rotation if it's a severe problem is to just cut out the peas and lentils as much as you can. There's 1 or 2 year, 2 or 3 year rotation definitely isn't going to cut it. So if you can go 5, 6, or as many years until the problem hopefully goes away. But I know their program's been looking at some different soil amendments and some different kind of cultivation type practices, but no hard and fast rules yet from what I hear.
Shama is also working on a soil test to sort of forecast the risk.
Okay.
So you could test your soil and say, you have a low risk or medium risk or high risk of aphanomyces in that field.
So if you are growing peas and lentils and you think there's a risk, is it developed yet, or they're working on it right now? So they're working on a tool so that you'd be able to submit a soil sample basically, and then assess your risk. So maybe you're growing a new piece that's down the street you've never grown on before. Maybe they had some problems in the past. They haven't kept good records, whatever, that kind of stuff. You should be able to get a good idea of whether or not your risk is going to be there or not. So, any more? Perfect. Thanks for questions.